Hello, today we are going to talk about plant versus animal cells. First, the plant cell. Here is a generalized plant cell that I have drawn, which includes 17 components to remember. And here's the cutaway view of the animal cell, also summarized into 17 components. Note that there are many differences, which we will talk about today. In this video, I will also be talking about the structure and function of these components. Here's just a reminder as I couldn't fit it on the page, but ER stands for endoplasmic reticulum, for both the rough ER and smooth ER. Okay, let's zoom into the nucleus part of the plant cell. It's important to understand the three components of the nucleus. This includes the nuclear envelope, the nucleolus, and chromatin. Okay, so here I have the nucleus on its own. We can see the three components clearly. And to make it easier to remember, I have this equation here which clearly shows that the nuclear envelope plus nucleolus plus chromatin equals the nucleus. So first we're going to look at the nuclear envelope. This is the blue colored double membrane in the image on the left. The nuclear envelope lines and encloses the nucleus from the cytoplasm. The cytoplasm is the fluid that is within the cell. And as the nuclear envelope is double membranous, there is an inner and outer membrane. And these layers are perforated by nuclear pores, meaning that the nuclear envelope is riddled with these tiny holes, and these are important for transportation. Overall, the nuclear envelope is continuous with the endoplasmic reticulum, meaning that the ER is physically connected to the nucleus by the outer membrane of this nuclear envelope. Okay, next we will look at the nucleolus. This is a non-membranous and very dense structure. It is involved in ribosome production, where these ribosomes will go on to help link amino acids together and form proteins. The nucleolus is made up of both RNA and proteins. Additionally, it contains one or more nucleoli. As we can see, it is spherical and the largest structure of the nucleus. Okay, so now chromatin is the final component we will look at. Chromatin consists of both DNA and proteins, and these are combined to form the chromosome found in the cells of higher organisms. Chromatin can be seen as individual condensed chromosomes. But it's important to note this is only visible in dividing cells. It's also important to know that the chromatins are highly organized condensed structures, so they are very dense. Alright, so here is the plant cell cutaway again, and now we're going to move on from the nucleus and look at the rough ER and the smooth ER. Here's the rough ER and smooth ER zoomed in on the drawing. And here's the entire structure. Remember, ER is the endoplasmic reticulum, and this structure surrounds the nucleus, including the components we just covered. That is the nuclear envelope, the nucleolus, and chromatin. Okay, so both the rough and smooth ER are a network of sheets and tubes that are membranous. Both the rough and smooth ER are active in the synthesis of membrane components. And these components are usually proteins and lipids. Specifically, the rough ER synthesizes proteins, while the smooth ER synthesizes lipids and steroids. Steroids are a type of lipid molecule made of 4 carbon rings. And so what makes a difference between rough ER and smooth ER? You might think it's the overall structure that makes the difference, like the smooth looks like a network of tubes, whilst the rough looks more flat. But actually it's about the presence of ribosomes. So as you can see, ribosomes are those yellow dots scattered on the rough ER. So, rough ER is ribosome studded, whereas smooth ER lacks ribosomes. Alright, moving on to what are ribosomes? Well, as already mentioned, they are these yellow dots on the rough ER. They are made from a combination of RNA and proteins, and these complexes are what make proteins in the cell. They make these proteins by reading messenger RNA and translate the mRNA genetic code into amino acids, and then finally to proteins. There are usually 3 locations for ribosomes. Either they freely move around in the cytosol, or like in this example, they are bound to the rough ER. And you can also see on the surface of the nucleus, they are bound to the nuclear envelope. Alright, back to the original diagram. So we are moving clockwise around the cell. We are now going to look at the central vacuole. That is this beige-colored empty space in the center of the plant cell. So the central vacuole. So this structure is only in plant cells and usually found in older plant cells. They are membrane bound. The functions include storage space, usually for water, but also nutrients too. Another important function is the breakdown of waste products. Typically, these are large molecules such as toxic chemicals and sometimes protein degradation by digestive enzymes too. The central vacuole also hydrolyzes waste through hydrolytic enzymes, including proteases, to break down proteins, nucleases, to break down nucleic acids, and glycosidases to break down carbohydrates. Lastly, it's important to note that an increase in the vacuole increases the plant growth. Usually, the central vacuole will apply pressure to the cell wall, causing it to expand. Okay, so in the plant cell, microtubules and microfilaments form the cytoskeleton of the cell. Note for later in this video that this is different for animal cells. The cytoskeleton reinforces and supports the cell's shape and organization of the different components. It also functions in cell movement. Usually this is the function of microfilaments only. As an example, microfilaments are involved with muscle contraction in animal cells. Down to a cellular level, microfilaments are involved with cell movement through the flagellum organelle. These components are made of proteins. Microtubules are made of tubulin proteins. So just remember, tubules are tubulin. And then obviously microfilaments are actin. And here, just for some perspective, microtubules are larger. That's why microtubules are often the structural component of larger complexes, for example, flagellum, which we will talk about later in this video. Chloroplasts. Okay, so these are the green rounded organelles in the drawing. They are only in plant cells. They are in the form of double membranous structures, as they are formoplastid. And these are photosynthetic organelles, hence why they are only in plant cells. This image at the top right shows the photosynthetic reaction that are vital for all plants, where carbon dioxide and water forms glucose and oxygen. Back to chloroplasts. As they are important for photosynthesis, just remember that their main function is to convert energy of sunlight to chemical energy stored in sugar molecules within the plants for their cell growth. Obviously, they are green in color, so they contain the chlorophyll pigments. Lastly, they are important for the biosynthesis of active compounds. This includes amino acids, fatty acids, and pigments, important for the growth and development of the implant. So next is plasmodesmata. These are also only found in plant cells. And in this image at the top right, they are shown as the purple colored channels connecting communication between the individual plant cells. So plasmodesmata are cytoplasmic channels in the cell wall. These channels function to connect the cytoplasms of the adjacent cells. Why do they do this? Well, in order for cell-to-cell communication. What does that involve? Well, it's the exchange of small molecules and macromolecules, such as nutrients and metabolites like sugar, protein, and messenger RNAs. On my diagram, it is shown as the yellow dots and the tunnel traveling through the adjacent cell wall and linking the plant cell walls and the adjacent cell walls together for viable communication. Alright, next is the adjacent cell wall. This is bound by the middle lamella, which overall is just a layer of material that helps plant cells join together. An easier way to remember the middle lamella is thinking of it as a glue for plant cell walls, just like my drawing here. And it's mostly composed of pectin, which is the same as the soluble fiber that is found in fruits. Although it's important to remember that it is also composed of other molecules such as calcium and magnesium pectates too. Alright, the cell wall. Again, this is only in plant cells. It is a physical barrier of cellulose, which is a type of carbohydrate. At the top of the screen here is my drawing of cellulose's structure. Here the drawing shows it's a linear polymer of 2 D-glucose units, linked by a beta 1 to 4 glycosidic bond. Anyways, this is just a side note for those interested in biochemistry. Back to biology. So as the cell wall is the outermost layer of the cell, it's important for maintaining the cell's shape and supporting its structure, but also protection from mechanical damage as well. And just to help you remember, the cell wall can be thought of as what makes plants stay upright, such as the bark of a tree. And as the cell wall is composed of cellulose, it's also the same carbohydrate that is in dietary fiber found in fruits and vegetables, and different types of cereals like wheat, rice, and corn. So the plasma membrane. This is the structure that encloses the cell. Although it's not the outermost layer, it is the layer beneath the cell wall, as the cell wall is attached to the outside of the plasma membrane. The plasma membrane is composed of a lipid bilayer. This is a thin membrane made of 2 layers of lipid molecules. As you can see in my drawing at the top right, and remember that the lipids are fats and oils, such as cholesterol, which is a part of the cell membrane. In this image, the heads of the lipid bilayer are hydrophilic, or water-loving, whereas the tails of the 2 fatty acid chains are hydrophobic, or water-fearing. What does this mean? Well, this means the plasma membrane is semi-permeable, and this is important for regulation and transport of materials. For example, small nonpolar, or that is non-charged molecules, such as O2, are soluble in the lipid bilayer and cross the plasma membrane freely, whereas larger uncharged polar molecules, such as glucose, are not able to. Okay, so peroxisomes. So these are membrane-bound organelles. They have metabolic functions, such as producing hydrogen peroxide as a byproduct when in reaction. And this byproduct of H2O2, that is hydrogen peroxide, is converted to H2O, which is obviously water. We can see this in my drawing below of the balanced reaction. Here the 2 hydrogen peroxide molecules break down into 2 water and 1 oxygen molecules. Okay, the mitochondrion or mitochondria. So mitochondrion is singular and mitochondria is plural. Mitochondrion is double membrane bound. Overall, it generates adenosine triphosphate, or ATP for short. And this ATP can be thought of the energy currency of the cell, where energy is stored and released. My drawing here shows the adenosine component of ATP derived from an adenine and ribose, where the triphosphate component is obvious in the name, 3-phosphate compounds. This high-energy bond in red is important, as this is what allows for ATP to function as the energy currency. And when broken by hydrolysis, this bond releases a significant amount of energy. Overall, mitochondria are involved with the aerobic cellular respiration process. This involves generating ATP from glucose using oxygen. So oxygen must be present for the aerobic processes to occur. So ATP is used as a way of storing the chemical energy produced by the mitochondria. And the ATP is produced by mitochondrial oxidative phosphorylation, where a series of chemical reactions involve electrons and protons. Okay, so that completes the plant cell portion of this video. Now, let's look at the animal cell and compare the differences. Here is the cutaway drawing of the animal cell to refresh our memory. So what doesn't the animal cell have? Well, there's no central vacuole, there's no chloroplast, no plasmodesmata, and no cell wall. So what's different in the animal cell compared to the plant cell? Well, there are lysosomes, there are microvilli, there are centrosomes, there are flagellum, and there are intermediate filaments. First, what is a lysosome? Well, these are membrane-bound organelles within the cytoplasm of the cell. They are digestive organelles, so they contain digestive enzymes. And the primary function is to hydrolyze macromolecules. So using water containing hydrolases, they break down molecules such as nucleic acids, proteins, and polysaccharides. Okay, microvilli. Now these are membrane projections at the base of the cell in this image. They increase the surface area of the cell, so these can help with absorption of nutrients. For example, microvilli increase the absorption of nutrients in the gut of animals and humans. Overall, they are supported by the actin cytoskeleton, which is another term for the microfilaments within the cell. Now, as you can see, they are thin, finger-like protrusions. Centrosomes. These are non-membranous. They contain a pair of centrioles that are connected and located in the center of the cell. And these are barrel-like in shape. They are involved in cell division and are the site where microtubules are initiated, meaning they are the start site for the assembly of microtubules. Next is flagellum. Flagellum are important for motility, or helping the cell move through liquids. They act as a sort of propeller made up of filament and a rotary motor. They are present in some animal cells, but not all animal cells. They are composed of a cluster of microtubules, and they are an extension of the plasma membrane. Lastly, we have intermediate filaments. These are in addition to microtubules and microfilaments. And these are components of the cytoskeleton. They reinforce the cell's shape and movement and are composed of mostly different kinds of protein subunits called keratins. Alright, so that completes this video. Let me know in the comments what other topics I should talk about next. Thanks for listening.